Great. Well, thanks so much, Andy, for the introduction, and really nice to meet everyone. I think this is a super exciting community, so excited to participate. I'm gonna go ahead and share my slides. Okay. All right. Does that work?
Yeah, we can see it.
Okay. Super. Wait, let me pull up the chat here. Okay, great. Yeah. So I'm James, and I just want to explore some of the directions we've been doing recently around this project that we call the Virtual Lab, where we basically try to create like a lab of AI agents to sort of mirror my physical lab at Stanford. So I'll say a little bit about how we built that and also share some of the discoveries that's made by the virtual lab. But before I get into the virtual lab parts, I first want to maybe advertise or announce this kind of an interesting experimental conference that we're organizing called Agents for Science. Um, and I also put in the link into the chat here. So this is kind of an interesting experiment where the authors and the reviewers for this conference are primarily AI agents, right? So basically, unlike all of the existing venues, right? So to participate in this conference, the first author of the paper should be an AI agent, right? So basically, we want AI to do most of the work, and the humans can be co-authors and providing advice to the AI agents. But the agents should be like doing the experiments and most of the writings of the paper. Right. And on the other side, we also have AI agents to do the reviews of these papers. Right. So all the papers will be reviewed by multiple AI reviewers. And then in the end, we'll have a set of human experts do sort of final assessment, the final selections of the papers that gets the top scores from the AI reviewers. And I think the motivation for this is that like the existing venues and journals, right, like NeurIPS or Nature, they do not allow AI as a co-author, right? So we want to flip this around, and so that for this our conference, you have to be an AI to submit, and then we'll also experiment using these AI reviewers, and everything will be public. So all the submissions and reviews will be public. So I think it'll be really interesting for us to see as a community, like how good is the AI agents at both writing papers and also reviewing papers, and also what are the strengths and limitations that we find by doing this. Right. So we're super excited about this experimental conference, and we'd love for folks here to participate. And we'll also have prizes for the top papers, which will be sort of generous compute credits for the top papers. Um, and we also actually have a partnership as a part of our conference. We have a partnership with this called the Dream Challenge, which is sort of a long-standing challenge where people actually provide experimental data to help to verify predictions, right? So the idea of this is that there's already a data set provided about predicting Alzheimer's disease from the from different RNA sequencing or gene expression data. And then people can train models on this data set and then submit their models' predictions. And then in the end, there will actually be like a new set of experimental data, that's sort of completely new data that's held out to verify those models. So similar to the flavors like the protein folding challenge by AlphaFold, but in this case for specifically for predicting Alzheimer's disease pathology. And what's new this year is that as a part of, in collaboration with our conference, we're also having an agentic track to this Dream Challenge, which means that for the first time now, you can also have AI agents submit predictions to this challenge. The agents' predictions will be evaluated the same way that we'll evaluate human experts' predictions. Right, so we'll have like also a leaderboard for the AI agents. So then we can actually have ground truth experiments to validate how well the agents can be able to predict and analyze Alzheimer's disease, right, from these genomics data. And if you participate in this challenge, then you can basically also have your agent write up a paper and submit it to the conference. So then it can be published in both places. Okay, great. Any questions about our conference? Okay, yeah. If not, I think it'll be a really fun conference, and I hope people would participate.
I have one question.
Oh yeah, please.
In Are there restrictions on like the role of humans in the pipeline here, or like is it supposed to be the whole end-to-end pipeline through an AI agent, or yeah, what are kind of the rules there?
Yeah, so we want this to be pretty inclusive because as we know, there are different ways of human researchers interacting with the agents. So our general guidelines that the AI agents should essentially play the role of like a first author of a standard research paper, right? So the agents should, do most of the work, and then the human co-authors can provide feedback to the agents, and then we'll have a checklist in the end where the submission for each submission, then the researchers can specify for each component, like for hypothesis generation, for data analysis, for interpretation, how much did they contribute, and also how much did the AI agent contribute. Right. So there's going to be some, I imagine, some heterogeneity, right, across the board. And when our conference is pretty inclusive, so we want people to submit, but then you can just capture that and in the checklist.
All
Awesome,
right.
that makes sense.
Yeah.
Yeah, we got a couple questions in the chat as well.
Yeah. Yeah. So I
First
see
one,
one.
go for it.
Oh, yeah. So one question is around, yes, we will be publishing the reviews and papers. So this will be done through open review. So the submissions, which are anonymous, but also the reviews will be public on open review. So similar setup as for example, you get with iClear and some of these other conferences on open review. Right. And I think this is actually be a good opportunity for the community to understand, like, if I look at reviews that are written by AI, like how good are these reviews? Do they find mistakes in the AI written papers and so on? There was also a question around if you can use like sort of chain prompts or tool use and so on, right? So yeah, so we're very inclusive. So it could be any sort of combinations of models or hybrid. You can use Cloud Code with different prompts. You can use your own customized AI agents that you built, right? Yeah, so you can try these different models across different stages. Super. Okay. So then for the main presentation, I want to explore a bit of this platform that we built called the Virtual Lab. It was recently published last week. So in this virtual lab, basically we created this platform where we have teams of different AI scientist agents with different expertise, right? And it's set up to sort of mirror my physical lab at Stanford. So in the virtual lab, there's an AI professor agent. And under the AI professor agent, there's actually a bunch of different AI students agents with expertise across different areas, like in computational biology, right, in chemistry, immunology, right? And they would actually have their own group meetings, right? So the AI professor would actually run their own group meetings, and the agents would actually meet together, and would give the agents some budget to do experiments, like real money to do experiments. So I'll just share a bit of how the agents interact, and also what are the kinds of experiments they're able to do, and some of the interesting scientific findings from the virtual lab. I'll also explain a bit of this system that we built called the Agent School, which is basically a mechanism where the agents can go to a school. So similar to how students can go to Stanford or other universities, the agents actually go to their own school and train themselves on how to become a deeper expert in these particular domains. So first, I'll explain how the interaction looks like in the virtual lab. So it's designed to be a human AI collaboration platform. So the way that we collaborate with the AI agents is that if I'm the human user, then I can mostly do this by talking to the AI PI or the AI professor agent. So, and I would mostly interact with the AI PI by explaining to them a high-level description of the kinds of projects I'm interested in. Right. So for example, I'll tell the AI PI I want to design some new binders to some potential drug candidates, right? Or maybe I say, okay, here's a really complex data set, help me analyze this data set, or maybe help me design a clinical trial, right? So it could be quite different kinds of projects. And then for each project, it's the job of the AI PI then to say, okay, for this project, what are the areas of expertise that are needed to tackle this problem? Right? If the project is to design new drug candidates, right? Maybe the PI would say it's useful to have, let's say, a protein engineering expert, right? Or it's useful to have a computational biology expert. Then it's the job of the PI agent then to create and then to train those sub-agents that have the relevant expertise. So for different projects, there could be different expertise that are needed. So that's why we make it quite flexible for the PI agent then to create different numbers of agents and different areas of expertise. I'll come back to in a couple of slides of how that creation and training of the agent happens. But for now, you can think of each agent as basically being like a base model. So it could be like a GPT-4, GPT-5, or Anthropic. You can use different base models. And then we'll have this additional layer of additional like fine-tuning and on top of these base models that sometimes is useful to endow these agents with additional expertise in these technical domains. So once the agents are created, then they will have their own group meetings, right? So similar to how we might have group meetings, the agents also run their own group meetings where they'll all meet together, and then the different agents can speak up sequentially to contribute ideas towards some research planning, research project. They also have one-on-one meetings, right? So similar to how I meet individually with my students, then the agents could also meet individually with the PI, the professor agent. And then what happens is one-on-one meetings that they will go over some subtasks like reviewing a piece of code or doing some particular analysis of a data set. Right? So they will alternate between these group meetings and one-on-one meetings. And then we equip the agents also with tools that are relevant for their expertise, right? So for example, we can equip like the protein engineering agent with access and usages of AlphaFold and other tools that are relevant. So in a sort of a sandbox environment, so they can safely execute and use those tools. So to make it a bit more concrete, so here's just an example of what's the a virtual lab group meeting look like, right? So in this example, this is actually the first project that we give to the virtual lab. So here we just ask them to tackle this quite challenging problem of designing potential therapeutics for the recent COVID variants, right? And here you can see that the PI agent spoke up first, right? So sort of lays the ground for the other agents, right? The PI agent says that, okay, our goal is to develop some new binders, right, to the latest COVID spike protein. And actually, at the time of this project, there were actually no known good binders to these recent COVID variants. So this is very much kind of a timely, but also very challenging, like open research problem that potentially would have substantial public health impact. And then the other agents can then speak up and contribute their ideas. Right? In this case, the immunologist agent spoke up next and actually recommended that to design nanobodies as the form of these potential binders for COVID. This is already kind of interesting and surprising because if you ask most human researchers, right, they would say, okay, let's design antibodies as binders because antibodies are much more common. You can find antibodies in most animals. And it's often used as sort of the binding substrate. But nanobodies are actually small cousins of antibodies. They're quite rare. They're only found in a few animals like camels, right? So it's kind of an orthodox suggestion to actually design nanobodies rather than the more common antibodies. But this actually turned out to be quite of a wise decision. So the machine learning agent spoke up next and says that, oh, it's actually a good idea to focus on nanobodies because this makes my job much easier as a machine learning agent, right? Because these nanobodies, since they're smaller structures, that's actually makes it easier for me to the ML agent to model their structures using deep learning models, right? I can get more accurate predictions. And then the computational biologist agent also says that, okay, if, in addition to using these deep learning models, let's also try to use these more physics-style simulations to generate more robust properties of these binders. And then we also have a critic agent that participates in all of these meetings whose job is to provide more skeptical feedback. In this case, the critic agent says that, well, if we're going to go down the path of designing nanobodies, then there's not a lot of public data around nanobodies for COVID, so we need to be super careful about overfitting. Right, so the actual meeting goes on for longer, but you can already see here I'm just showing basically the transcript from the first part of the meeting, right? And you can already see there's some interesting discussions and how the agents can build upon each other. Now there's a question, interesting question around like how do we determine the sequence of which agents speak, right? Which is a good question. So here, what we do find is that it depends on which agent speaks first or second. You can actually arrive at different ideas, right? So one of the nice things with the virtual labs is that we can actually run multiple meetings in parallel, so that we can actually in parallel meetings have different agents speak in different orders. And this is possible because and this is something I'm very envious of the virtual lab, is that their meetings are so much more efficient than my meetings, right? So each of their meetings maybe takes a few seconds or a minute. So in the time that I had one of my meetings, they already had hundreds of their discussions. And since the agents here, they don't get tired, so they actually run multiple meetings in parallel. So basically for every scientific decision they want to discuss, they actually run it 5 times. And in the parallel meetings, you can have different agents speak in different orders, so you can have different randomness, right? So they actually come up with different ideas. And then what we do is to basically have the professor agent to read the transcripts of all these parallel meetings and to come to a consensus of what to do next, right? So it's kind of an effective way to explore a broader range of ideas, right? But still have some sort of decision maker to decide what to do next. And in case you're curious, I'm showing this pie chart, basically how often the different participants would speak in these virtual lab discussions. So for example, the human researchers, so for example, myself, my students, we don't talk very much, right? So maybe we talk about 1% of the time. So our job is to mostly provide high-level guidance to the agents. For example, we tell the agents, right, what problems are important to us, like, designing good COVID therapeutics is important to us and, important to human societies, right? And we also tell the agents what are some of the constraints, right? Like we tell them, for example, here's how much money we have to do the wet lab experiments, so don't go too crazy on that. Right? But mostly we leave it to the agents on coming up with their own experimental designs and then execute on those, right? We don't want to micromanage the AI scientists too much. So the PI agent speaks about 20% of the time, right? So the job of the PI agent is to mostly like organize these meetings, right? So it is kind of a little bit of a hierarchical structure in that then the PI agent here sort of makes the final decision at every step, right? It would take into consideration like the suggestions from all these other agents, right? And the PI agent will make a final decision. Then the computational biologist agent, in this case, does the most work because this agent has to design basically the overall workflow for designing these nanobodies. Right? So does the most talking. The machine learning agent's job is to actually basically write all the code for all the other agents. Right? So here it doesn't talk too much in English, but also speaks quite a bit, mostly like in terms of code. It writes the code and implementations for the ideas proposed by all the other agents. Okay. Any question about how the agent interaction looks like? Yeah, there's a question around, is there a review step to check for quality? Yes, so that's basically the role of the scientific critic, which is to act as a reviewer in all of the meetings. So the critic basically would join all the group meetings and also the individual meetings. So for example, if the machine learning agent writes the code, then it's the job of this reviewer or the critic agent to basically go through and then verify and run unit tests and debug the code.
I think, Richard, did you have a question?
Yeah, but I think my question has already been like answered. So I was like asking about like the order of which agent like should talk, or like maybe I was thinking maybe if you let someone talk first, then it gives like some sort of like more weights to that, because then the whole idea is biased to that like initial point or something. So maybe just like a follow-up question. So do you think if you do these parallel meetings, like do you still some find some instances where like although you randomize these things, but you still find some bias towards certain like speakers or something? Yeah.
Yes, that's a good question. So sometimes one of the parallel meetings might go off rails into some directions. Right? So we do find that one of the benefits of parallel meetings is that like the consensus tends to be much more robust and have less bias and less variance compared to the individual meetings. And I think you're also getting to a good point in that I think there's an interesting social dynamic that emerges among the agents, right? So who speaks first and who talks more than others, that also leads to the kinds of science that's being done, right? And I think it's the same with human labs, right? So in your own group meetings, some people are more talkative than others, right? And that affects sort of the decisions.
Yeah.
I think something that's very interesting that I'm very interested in, but that's easy to do with the agents, but it's impossible to do with humans, is that we could do all sorts of counterfactuals, right? Like we could run a parallel meeting and say, okay, what if, let's say the machine learning agent speaks first, right? Or what if you can't talk, right? What if we give the agents different personalities, right? So we can actually do all of these sort of like a metaverse of all these counterfactuals and see how does that affect the science that's been done. Like we have done experiments to say, okay, what happens if we have more scientists that participate in the meetings, increase the size of the virtual lab, and sometimes that actually hurts the performance. Right? It's very hard to do those experiments with human labs, but we can actually do that systematically with the virtual lab as a way to basically figure out what is the optimal meeting structure, right, discussion structure that can lead to the most creative and innovative science.
Yeah, right. That makes sense. Thank you.
Yeah. So there's a question from John around how do we evaluate the quality of a scientific idea's outputs? So it's an important question. So ultimately, what we'll do here is to actually do wet lab experiments to validate the discussions from the virtual lab. I think that's really the best oracle, right? The ground truth is that we have to do the experiment to validate. Well, and I'll show you in a couple of slides the experimental results. So the next part I want to briefly mention is to say a little bit more of how we can actually improve these agents, right? So initially the agents are we can use sort of the foundation models like the GPT models or any of your favorite foundation models, language models. While these models are quite good at broad knowledge, right? If you go really deep into a particular technical domain, right? Sometimes the models do not have sufficient information or reasoning about that domain. So, for example, if you say, okay, so what are the latest developments, right, in COVID clinical trials, right? The model does not have that up-to-date information. Or if there's like a new tool, like, AlphaFold 3, right? Then the models may not be familiar with how to use that new tool very well. So we experimented with different strategies for improving these agents. So that's part of what we call this agent school. So here's how the school works, or briefly. So basically, we first tell the agents here are some of the topics that are important, right? Maybe one topic could be, learn about the most recent COVID variants, right? And then for each of those topics, then it's the job of the agent to basically design their own learning curriculum. And they do this by coming up with a set of questions that are relevant for that topic. And we equip the agents with the ability to search and find papers, for example, from PubMed and other resources. So based on these questions, the agents actually go out and download relevant papers. They'll filter for papers that are likely to be relevant and high quality. And then they can read those papers, right? As the agents read those papers, we also experiment with different fine-tuning mechanisms so that the agents can then start to update their model parameters as they're reading these papers to help them to ingest additional new insights, new information. And it would not be a very good school without some sort of assessment mechanism. So we basically also have other agents, sort of like teacher agents, who would also read textbooks and these recent papers and use that to design QAs, right? Basically design quizzes. And the student agents then would have to do well on those quizzes before they pass, right? If they don't do well, they have to go back to school, find it, download, read additional papers, do additional training until they can pass these assessments. So it's a relatively autonomous process whereby we decided on the topics, humans decided on the topics, but then the agents basically decide their own learning curriculum and they do their own retrievals and do their own updates to learn about those topics. So this enables the agents to basically learn about topics, even in areas that I have no expertise in, right? So I wouldn't be able to teach the agents very well around certain areas of chemistry, but the agents can still be an expert there because they can do their own learning curriculums. And here, there's a question around now, what happens to papers that are behind paywalls? So currently, we mostly allow the agents to basically download papers from PubMed Central, which are our open access papers, so they can get to the full text. For the paywall ones, then they can get like often the abstracts, so they can still learn something about the app from the abstracts. But we prefer if the agents actually learn from the full text from the open access articles. Great. So now that we have a sense of how the agents interact, and then how the schooling happens, right? So I want to come back to this case study that we started off with, right, of the agents designing these potential therapeutics and binders for the COVID variants. So recall that their goal is to design binders that can bind to the spike protein of the COVID, and it's important to have these binders because often in order to have a good therapeutic or good vaccine, they need to have something that binds to the target virus. And the agents decided to design nanobodies, right? The binders in the form of nanobodies. And to do this, they said, okay, so it's useful to use several tools to help them to design these nanobodies. So the agents actually decided to use 3 quite sophisticated tools called ESM, AlphaFold, and Rosetta. And then they will actually put these tools into a new computational workflow that they could then run in silico to help them to optimize for these nanobodies. So here's how the workflow works. This is the workflow that's designed and implemented by the virtual lab agents. So they first decided to use this protein language model called ESM. ESM basically will take as input some amino acid sequence and just tell you how likely is the sequence to form like a stable functional protein, right? So basically computes a score, right? So the agent will first use that to filter for likely candidates. And then they will pass those candidate nanobodies into AlphaFold together with the spike protein, right? So then the AlphaFold will generate this 3-dimensional structure of how the nanobody actually binds to that target spike protein, right? And the agent will actually look at that 3-dimensional structure to see how well that looks, and then we'll pass that 3-dimensional structure into Rosetta, which does a more like a physics-based simulation. It will take the 3-dimensional structure from AlphaFold, actually relax the proteins, and then rebind them, right? So it estimates the binding affinity between the nanobody and then the target, right? So based on looking at all 3 of these, the outputs from all 3 tools, then the agents will do like a holistic reasoning and assessment to see how likely is this to be a good candidate binder. And then we'll iterate through this process multiple rounds to come up with the most promising candidates. Right, so the space of potential nanobodies is just astronomically large, right? There are trillions and trillions of potential nanobodies that the agents could design. But by using these tools, and the agents are able to actually filter down to a small set of the most promising ones. All right, so this workflow is actually quite interesting because it's different from what people have published before, right? So we looked at all these previous papers. Some people often would use maybe 1 or 2 of these tools. But to our knowledge, it's actually the first time that there's this computational workflow that uses ESM, AlphaFold, and Rosetta in this way together, right? And moreover, the agents are not limited to using existing tools. They actually made modifications to these tools, right? So for example, they took this protein language model ESM. So previous papers by human researchers had some particular ways of extracting embeddings and scores from this ESM model. So agents actually decided to change that and had a different scoring mechanism that, in retrospect, also made a lot of sense to us. So they're able to take tools, modify these tools in interesting ways, right, to adapt it to their use case, and then also combine these different tools into interesting new workflows to help them to solve these quite open-ended problem. So in this case, now we gave the agents enough budget, so they designed 92 nanobodies in silico computationally. But they can't do their own experiments yet, right? So then basically we, with our collaborators, we made each of these 92 nanobodies in the real labs, and then we tested them to see how well do they bind to the COVID variants in these real wet lab experiments. So here I'm showing 2 example nanobody proteins designed by the virtual lab agents, right? These are entirely new nanobodies that have not been seen before. And the bottom here basically shows the binding affinities, right? So I'll just parse through and explain each of these curves one by one. So if we look at this bottom red curve was the open red circles. So that's where if we take the human designed nanobodies, right, existing nanobodies, and see how well do they bind to the recent COVID variants. This is called the JN1 variant that started to spread last year. So you can see that existing human designed nanobodies basically show no binding to this recent COVID variant. And then the solid red curve is basically the nanobodies designed by the virtual lab agents. So it does show now more promising binding signals, right, to this recent variant. And then the 2 blue curves basically show if we take these 2 nanobodies and test them against a very different strain of COVID, basically from Wuhan from 5 years ago, right? So the nanobody designed by the virtual lab, which is the solid blue curve, shows even more enhanced binding to this very different Wuhan strain of the virus. And the reason why that's quite exciting is that ideally you want to have the same binder that can bind even as the virus evolves over time, right? And here the virus has evolved quite a bit from Wuhan to last year, but now the nanobody designed by the agents actually does show, seems like consistent binding across this broad spectrum. So that's quite exciting. What's not shown here, but we did additional experiment to test how well these nanobodies actually bind to host proteins, and they do not bind to human host proteins. And also, they do not bind to other viruses, which is also important because you don't want these therapeutics or vaccines to bind to your own proteins in order to reduce potential side effects and toxicity, right? So in other words, the proteins binders designed by the virtual lab, they show specificity, right? But within the COVID family, this seems to show quite broad spectrum binding. So there's definitely more experiments that needs to be done to further improve and validate these designs. But I think this is already quite exciting because just within basically a couple of days, the virtual lab is able to come up with a research plan, design the computational workflow, implement it, and execute the workflow, and help us to come up with these candidate antibodies. The wet lab experiments took much longer, right? It took a few months, right? But the computational part was actually greatly accelerated, and that part is really exciting. So maybe just to wrap up this discussion here, right? So we designed the virtual lab to actually be quite versatile platform. So it's all open source actually, so people here are welcome to try it, right? So it's not limited to designing protein binders. So you can use them to design other kind of scientific applications. Could be even in outside of the life sciences, right? So people can use this for like studying quantum computing, right, or material discovery and so on. We have done experiments here in the paper to show that I think actually these teams of multiple agents with different expertise ends up working much better than having a single agent that try to reason through everything and do all of the process. I think one reason for this is that these multiple agents in this virtual lab interactions, often they have debates and disagreements, right? When the agents have disagreements, right, maybe the one agent disagree with another agent, they have to come to some resolutions, and often in those discussions or resolutions, that's where it gets into really interesting in-depth scientific reasoning. Here, we found to be quite helpful to provide these agents with additional resources, so they should have some memory components so they can actually remember what did they discuss in the previous group meetings or one-on-one meetings. Right? We also provide them with sort of like a sandbox so they can actually execute these tools like AlphaFold in a safe environment. And I think the other thing that's worth noting is that these agents, they do have their own interesting social dynamics. So we have been discussing, depends on which agents speak first. Sometimes agents talk more than others, and that can all lead to different kinds of creativity and different solutions. Right? And one of the nice things with the virtual lab setups that we can actually run multiple these lab meetings in parallel, so we can actually explore different dynamics and almost basically having like a social science petri dish, right, to understand what are the kinds of interactions that seems to be the most conducive for scientific innovations. So I'll stop here. And just to also plug this again, like you could, for example, use the virtual lab to help you to write a paper, right? And then you can submit the paper to our new conference, right? Where it will be reviewed by other AI agents, right? So that's also perfectly a good use case. Right. So I'll stop here and happy to take any further questions.